Alright folks, let's talk about enzymes. Think of them as tiny biological machines, protein machines to be exact. They are catalysts. They speed up chemical reactions. Without them, life and food would be very slow. Now why should you, a future food safety officer, care? Because enzymes are everywhere in food. They make cheese cheesy, they make bread rise, they can also make apples turn brown, or make milk go bad. See? They can be good guys, or they can be the villains in your food safety story. Understanding them is key. If you understand how enzymes work, you can control them. This means better food quality, and that's your job, right? Knowledge is your best tool. To understand enzymes, we need to organize them. The International Union of Biochemistry and Molecular Biology, or IUBMB, gave us a system. They sorted enzymes into 6 main classes. Think of it like a big family tree. Each class has a specific job description. Let's meet the first 3. First up, class number 1, oxidoreductases. These enzymes handle oxidation and reduction reactions. When an apple turns brown after you slice it, that's an oxidoreductase, polyphenol oxidase, at work. Next we have class number 2, transferases. These enzymes are like molecular delivery trucks. They transfer functional groups from one molecule to another. Super important in many biological pathways. And rounding out our first trio, class number 3, hydrolysis. Hydrolyses use water to break chemical bonds. Digestion relies heavily on hydrolyses. They break down big food molecules, like proteins, carbohydrates, and fats, into smaller pieces our bodies can use, or that microbes can use to spoil food. Alright, we've met half of the enzyme big bosses, but the IUBMB family has 6 members in total. Let's get acquainted with the remaining 3. Class number 4, lyases. These enzymes are bond breakers. They cleave various chemical bonds, or they can do the reverse, adding groups to double bonds. Next up, class number 5, isomerases. Isomers are molecules with the same chemical formula, but they have a different arrangement of atoms. Isomerases rearrange atoms within a single molecule, converting one isomer into another. This can dramatically change the molecule's properties. Finally, we have class number 6, ligases. Ligases are all about joining things together. They catalyze the formation of new chemical bonds, joining 2 molecules, often larger ones. This process usually requires energy, essential for building complex structures in cells. So, we know enzymes are protein catalysts, and we know they come in different classes, but how do they actually their jobs? It's not magic, folks, it's chemistry. Let's peek under the hood. We need to talk about the active site. This is where all the action happens. Every enzyme has a special little pocket or groove. This is called the active site. Only certain molecules called substrates can fit in, like a key fitting into a lock. When the substrate binds to the active site, they form something called an enzyme-substrate complex. It's a temporary partnership. During this meeting, the enzyme gets to work. It might bend the substrate, or stress its chemical bonds, or bring reactive groups close together. Once the reaction is complete, the substrate has been changed into products. The enzyme then releases these products. The enzyme itself is unchanged. Enzymes are powerful, yes, but they are also a bit sensitive. Various factors can affect their activity, speed them up, slow them down, or stop them completely. As food safety officers, these are your control dials. Temperature is a big one. Most enzymes have an optimal temperature. If it's too cold, enzymes slow down. This is why we refrigerate food. If it gets too hot, enzymes can denature. That means they lose their specific 3-dimensional shape. No shape, no function. Next up is pH. Enzymes also have an optimal pH range. If the pH is too far from the optimum, the enzyme structure can change. Its activity will decrease or stop altogether. Substrate concentration also plays a role. Generally, more substrate means a faster reaction, but only until the enzymes are saturated. Now, enzymes aren't just about spoilage or problems. Humans have been cleverly using enzymes in food processing for centuries. Today, we harness specific enzymes to make food tastier, last longer, or look better. Let's look at some delicious examples. First stop, the bakery. Amylases are stars in the baking world. These enzymes break down starch. Starch is a big, complex carbohydrate. Amylases chop it into smaller sugars. Yeast loves these simple sugars. Yeast eats sugar, produces carbon dioxide. That gas makes bread rise, light and fluffy. Next, let's talk cheese. Proteases are key players here. In cheesemaking, an enzyme preparation called rennet is often used. Rennet contains a protease called chymosin. Chymosin specifically targets a milk protein, casein. It causes the milk to curdle, separating into solid curds in liquid whey. So far, we've talked about adding enzymes or controlling the ones already there, but what about the enzymes that are naturally present in food? These are called endogenous enzymes. They are part of the food's original makeup. Let's explore some of these natural food modifiers. Think about a banana. It starts green, hard, and not very sweet. Over time, it ripens. It becomes yellow, soft, and deliciously sweet. What's happening there? Endogenous enzymes. Amylases are breaking down starches into sugars. Pectinases are softening the fruit's texture. Other enzymes are developing those characteristic flavors and aromas. This ripening process is a complex enzymatic concert. Now for a less appealing but common phenomenon: enzymatic browning. Slice an apple, a potato, or an avocado. Leave it out for a bit, it starts to turn brown. The main culprit here is an enzyme, usually polyphenol oxidase, PPO. When you cut the fruit or vegetable, cells are damaged. Well folks, we've been on quite a journey through the microscopic world of enzymes. These protein powerhouses are truly amazing. Let's quickly recap what we've learned. Enzymes are biological catalysts. They speed up reactions in food. They are essential for life, and incredibly important in food science and safety. They are the tiny gears that make the food world turn. We learned about the IUBMB classification, those 6 big classes: oxidoreductases, transferases, hydrolyses, and lyases, isomerases, ligases. Knowing these helps you understand an enzyme's job. We also peeked at enzyme kinetics, the active site, the enzyme substrate complex, and how factors like temperature and pH are crucial. This knowledge isn't just for show, it's for action. It empowers you to make informed decisions. Understanding enzymes is not just academic, it's profoundly practical.